Before starting, I'd like to go over a few things. So as stated in the email invitation, today's presentation will be in English, and the presentation slides in French and English, and the recorded webinar from today will be sent to all participants by email next week. Tel qu'indiqué dans le courriel d'invitation, la présentation sera donnée en anglais et en français, mais la présentation sera disponible pour vous en français et en anglais, et l'enregistrement du webinaire d'aujourd'hui sera envoyé à tous les participants par courriel la semaine prochaine. Nous vous invitons d'utiliser l'icône de questions au bas de votre écran pour vos questions. Les questions peuvent être posées soit en anglais ou en français, mais nous allons attendre à la fin de la présentation pour répondre aux questions. So we also use the Q&A button at the bottom of your screen to ask your questions. So they may be asked in English or French, but we'll wait until the end of the presentation to go over the answers to answer your questions. Next slide. Some of you may have participated in our past webinars. They are available on YouTube, and our next one, Forecasting Fall Crops, is scheduled for July 12th. So make sure to sign up to our newsletter to receive information on the fall webinar series and topics. And before we get started, I'd like to acknowledge that we conduct our work activities on the treaty lands and territory of numerous and diverse Indigenous nations and pay tribute to their heritage and legacy. Indigenous nations, I apologize. We aim to walk lightly, harvest with respect, and learn from local knowledge keepers of every nation. I'd like to pass it on now to Mary Knockwood. Mary is the Indigenous Program Coordinator for the National Tree Seed Center. Mary?
Sorry, I had to unmute. Hi, I'm Mary Knockwood. I'm an Indigenous woman from the Fort Folly First Nation, which is the smallest Indigenous community in the Atlantic. I am working for the Indigenous programs for the Atlantic Forestry Center, and I'll be working on the Indigenous seed collections moving forward. So that's me. Okay, so in February of this year, the National Tree Seed Center staff compared the research done by the Q-Seed Information Database and a research paper written by Appert et al. And it was found that 43% of Canadian boreal species valued by Indigenous people lack seed research. I think this is extremely important to keep in the backs of our minds as we progress with the Indigenous Seed Collections Program. This Indigenous Seed Collections Program looks to include Indigenous values and ethical harvesting and considerations. What does that look like? Making changes in operations to include Indigenous values, learning from Indigenous groups we work with, learning about what you know about the species in your territories, understanding what you value. All of this so that we may start the Indigenous Seed Collections Program on the right foot by learning, listening, and sharing what we know and have learned, and incorporating Indigenous values into the National Tree Seed Center's practices. Sharing the work. This is a national program, so it's looking at Canada as a whole, and there are over 600 distinct First Nations, Inuit, and Métis, which encompasses more than 60 languages with different practices and traditions. How do we ensure that we are inclusive of all Indigenous groups across Canada? That's something that we're going to endeavor to try to make happen. Next. The National Tree Seed Center can support Indigenous communities, groups, and organizations in seed collections through providing training, storage, and germination testing of their seeds. We can help in finding potential funding sources, and we can ensure the protection of your intellectual property rights along with your seeds through the development of memorandums of understanding. Next. And we will work together. We will work together with you in developing opportunities for networking, learning, and sharing. Our door is always open. If you wish to visit, to see what we do, or to share your experiences with us, please drop by. We're always welcoming anybody to come and see us. We're looking at linking Indigenous groups, communities, and organizations with the many different NRCan scientists that are working on subjects that may be of interest or concern for you, your organizations, your groups, or your communities. And most importantly, we want to listen, learn, and grow together to build a strong Indigenous seed collections program. Next, Donnie, that's you.
Hey, well, thanks, Mary. And again, we thought it was a good opportunity to introduce our Indigenous seed collection program at this time before we head into the presentation for the day, which is mainly on collection of our berry crops. Before we get into that, though, again, just this one slide to talk about the National Tree Seed Center and an evolving mandate. And again, if you've attended our previous seminars, I apologize for the repetition. There will be very limited repetition, but here I go. So again, the National Tree Seed Center, it was established in 1967 in Petawawa, Ontario. And at that time, it was established to focus on obtaining, storing, and providing native Canadian tree seed of known origin for research. And it was during these early days and years that the Canadian Forest Service, along with the National Tree Seed Center, worked collaboratively with the provincial seed centers across the country to develop the tree breeding program that is still in place today. And much of the seed currently used in our industrial sector comes from these well-established programs. In 1996, the Seed Center moved to Fredericton, New Brunswick, and this coincided with new commitments by our federal government to the Convention of Biodiversity and the Federal Species at Risk Act. So it was at this time that the mandate of the center grew to include shrubs and the conservation of genetic resources under threat by invasive pests, pathogens, and climate change. Although climate change wasn't really a thing in 1996, it was really not until the early 2000s where that term started coming about. But over the 50-year history of the Seed Center, we have gained expertise in the collection and distribution of seed for research and in collaborating on conservation and recovery programs, along with innovations in small-scale seed handling, testing, and storage protocols. So now, with the announcement of the 2 Billion Tree Program, we envision that there may be potential seed-related gaps regarding the resources and training materials available to partners of the program. So again, this presentation that we're giving in this seminar series, although it's funded by the 2 Billion Tree Program, we want to stress that you don't have to be part of the program to partake in these seminars or partake in any of our services. We also wanted to recognize that different regions in the countries will have varying degrees of expertise and support available. And all we're trying to do is work with the organizations in these regions and then fill gaps where they exist across the country. And that's really what has led us to putting on this seminar series. So we can move to the next slide, please. And this brings us to our first set of polling questions. Again, by having you answer these polling questions, it really helps us identify the best way to support you moving forward. We don't want to make these seminars about us telling you what we think you need. We really want to gear them towards what you need. And hopefully you'll see that in this presentation that we're doing today, that we've really taken cues from our initial seminars and put them into the rest of this presentation. So there's 3 total polling questions we're going to put up here, and we're going to give you, 30 seconds at each one. Good, so there's our answers for our first polling question. Our second polling question will come up here, and it's, are you considering planting shrubs in your 2 billion tree projects? Good number of yeses, depending on the planting site, great. And then our third polling question will come up here. Are you planning to hire or contract additional seed collectors this year? While you're answering that, I just also wanted to say that the last seminar, we made an announcement that the 2 Billion Tree program is working on exploring potential call proposals to include seed collecting as part of the 2 Billion Tree program, and that was fairly new. And I just wanted to reinstate that since our last presentation, this is indeed continuing to progress in a positive direction. Good. Well, thank you very much for answering those questions. Again, that will help provide information back to the 2 Billion Tree team and also help us as we move forward in our presentations. So we can go to the next slide, please. Good. So on this slide, we're simply trying to provide the definitions that came out of discussions with the 2 Billion Tree call proposals back in December 2021. So there were lots of questions about what is a shrub, what is a tree, because when the program was initially announced, shrubs were not part of it. But somewhere over the course of time and through input from people interested in the program, this was changed to include. So at this time, the 2 Billion Tree program will cover shrub species up to 15% of the total project costs, providing that shrubs do not outnumber trees in your proposals. Yeah, so I think we can move on. So again, a lot of this seminar is about providing you with information, hoping that you will download and print off the presentation and then take some more of this information in afterwards. So we're trying to give you as much information as we can, hoping that it'll move you in the long run. So when you are, first of all, if and when you are applying for funding through the 2 Billion Tree program, it helps to know what are the benefits, because again, if you're going to include shrubs, and we're talking about shrubs here because a lot of our fruit-bearing trees there are fruit-bearing trees, but they are trees and shrubs, and a lot of times people will classify them as shrubs when indeed some of them are quite large. A lot of our cherries and other species can be quite large. So while the 2 Billion Tree program, you know, it's about carbon sequestration, it really is about a lot more, and this includes a wide range of ecosystem services. So again, when you look at this list, there are many situations when planting and therefore collecting seed from shrub species and berry-bearing trees, it's a very viable option. So if you're thinking about collecting seeds from these types of species as part of your proposals, these are just a few trait-based potential benefits to consider for your proposals. And we can move on again. So if you participated in our first seminars, we talked a lot about seed source, and we stressed the importance of seed source for all of our tree seed collections. We know that using appropriate seed source for trees matters, and our forest industries and reclamation have shown this. They have the science that backs this up, that if we're not using the appropriate seed source, then it costs money, mainly because we don't get the growth and the survival rates that we're looking for. And so we really want to be working with natural selection and adaptive trait principles and not in opposition. But the question comes up, well, is this important for our shrub species? And that's a really good question. And the answer is that although we don't have the science to prove this at this time, and mainly because they're just understudied, one would have to anticipate that it would be very similar to our tree species. And I'm really pleased to say that seed source identification has been put as being very important as part of the 2 Billion Tree program. And if it's important for the trees, it's going to be considered also for our shrub species. So if you are collecting shrub species and you want them to be to be usable as part of the 2 Billion Tree program, it's really important that you start geo-referencing your seed locations where you're making your collections from, so that they are eligible for the program. And that's something that probably hasn't been done at a very high rate. But again, if you want your collections to be part of the program, we really need to start geo-referencing things now. And we can move on. If you are considering working with collecting fleshy fruit species, what are some of the issues and bottlenecks? And we have a list of them here. So many of them simply do not produce prolific amounts of seed compared to a conifer, poplar, and birch. So again, we talked last one with a poplar catkin, you can get up to 1,000 seed from one catkin. That's not going to be the case with your berry collection. So it's very labor intensive. You know, one seed or one berry in many times equals one seed. You know, one of the other issues we're definitely seeing is decline in pollinators and fragmented populations and habitats, which leads to poor pollination and also inbreeding depression. So a lot of times we'll see the fruit, but the seed within that fruit can be hollow, empty because it didn't get pollinated or due to inbreeding. Another thing you're really going to see when you're out making your fleshy fruit collections are bird predators and rapid dispersal. So a lot of times, as soon as these seeds are ready, the sugar content is very high in the berry, and the birds are just waiting for that. So they can clean off, you know, a massive stand or a whole ecosystem off their berries in a very, very rapid time. So you really have to be ready to make your collections. And that's very similar to the spring seeders that we talked about in the last presentation. The other thing you have to consider is the size of the berries. Again, so if we're talking about one berry, one seed, think about the weight of that seed. So there's a lot more weight, a lot more volume. So in your potential proposals that could be coming up for seed collection, you really think about the added costs and planning for shipping, collection, and all those kind of factors. And then another issue is germination issues. You know, with a lot of our spring seeders, you know, we expect 90 to 100% germination. We might not get that with a lot of our berries, and for a lot of our berry species, we simply don't have all the protocols worked out. And a lot of that has to do with a lot of our berries, they're ingested, and it's a digestive enzymes of the gut of whatever animal has ingested them that helps break down that seed coat. And a lot of time that happens before the seed goes into stratification. So for a lot of species, we're still working out. We know by doing cut tests that we have potential good, viable, healthy seed in that seed coat, but it's about how do we break dormancy and how do we store them properly so that we get a higher percentage of germinants when we want them. So we can move to the next slide, please. So although shrubs are often easier to start collecting because they're reachable, there are still many other factors that inhibit good collections. And on this slide, we're simply providing a list of factors and resources that one could and potentially should consider if you are getting into berry collections. And these are references here that we commonly use, including in Ontario, and this Seeds of Ontario, Trees and Shrubs provided by the FGC in Ontario. We find this is not good Morrison edition Ontario, but we use it for a lot of our Maritimes and Quebec species, and even sort of in the lower prairies. The NATE website is listed down here below. So again, in the Northern Alberta Institute of Technology, and then there's also the publications by Ann Sumac here for the SNR Oil Sands Research Information Network. So a lot of really good information in all of these manuals and books. And again, we can provide links for those at the end of this presentation. And we can move to the next slide, please. So something else, it's what are the tools you need when it comes to berry collections? And some of the tools are a little bit different than what you'd be using, like no pole pruners in general are needed. But again, at the end of the presentation, we're going to provide a document that will have a list of all the tools that we use. And again, if you're listening in and you have specific tools that you don't see in that list, please feel free to share. Some of the things that you really have to be aware of is expert identification. What you don't want to be doing is going out and think you're collecting one thing and it's something else. So early in the summer, we're talking July, really good idea to get out there with your identification keys. Once you find nice bushes, or trees and shrubs with a high berry count, is really get out there and make sure you're doing your identification to make sure you know what you're collecting. Then doing cut tests right away. So we use dog nail clippers. They're really good for berries for doing cut tests to them. Again, what you don't want to do is sort of earmark collection points only to go back in the late summer to find out that all those berries are hollow or empty. So the best time to start doing your cut tests is when you're out there doing your forecasting. And again, everything else on here, again, will provide lists. And again, one thing we tend to do when we're out doing our forecasting is really pinpointing many different sites with high crop potential. Again, we don't want to put all our eggs in one basket, let's say. Make sure you have lots of sites for potential picking, because again, there's lots of factors working against you, including animal predation, heavy storms can come along, certain areas, high winds that can blow all your seed off before they're completely ripe. So again, just make sure you have lots of locations when you're getting into berry collecting. Go to the next slide, please. Again, finding those large populations in bumper crop years are really important. Getting out and scouting right after flowering. And then, as I was saying, looking beforehand for color changes. Even before color changes. So again, here we have, I believe this is black cherry here. The berries are nice and green. So if you did a cut test on this particular seed here, it looks pretty early in the season. The seed inside is not going to be this nice dark brown. It's probably going to be a pale yellow to white. So that's another indicator of when the berries are ready to collect. Excuse me. And again, on the next slide here coming up, we can put the next slide too, because it kind of talks about this here. But we like to say there's no real guessworking when it comes to collections of mature seed. There are going to be color indicators, and you can either ignore what the seed is telling you and collect it too early when they're green and have seed that's not viable. Or if you wait too long, you're going to lose everything, especially for the berry crops, because there are so many predators and bird species out there just waiting for the seed to change its flavor. So again, going on those visual cues. And if for some reason the color of mature seed is not known, so this is a nice one here of black cherry. So again, certain berries turn red like the mountain ash, but for black cherry, you might think, oh, they were green and they turned red, when we're actually waiting for them to turn black. Again, if you cut open this seed, you would probably see that the seed within it, sorry, if you cut open the fruit, the seed within it hasn't turned that nice brown color. The other indicator, of course, is the berries will pull really easily off the stems once they are ready. So again, if you're out there collecting and you're really tugging at your seed, more than likely they're not ripe. And you can collect some of the berries a little bit early, but their potential for long-term storage is missing. So one of the things with most seed species, is that most tree seed species, is that the last thing the seed inherits from its parent is that ability to store long-term. So that's just a real quick overview of the forecasting and what you're looking for before you collect. And I'm going to hand things over to one of our technicians, Kieran Volt, who's actually going to go into a little bit more of the collection and processing and lab ordeals. Thank you.
Yeah, hello, everyone. So as Donnie was saying, a very important part of seed collecting is to document where you collected the seed from. And we are very careful about that at the Seed Center, and we encourage anyone who is collecting seed to also be very careful. So we generally put the information in multiple places. We will, and when we're collecting berries, we always put in plastic bags. So we'll write who collected it, when we collected it, and where we collected it on the bag. And we then will also write on a tag, which is on the right top right-hand corner there on the slide. And we'll put that information in the bag as well. Now, it's most advisable to write it in pencil because the berries can be wet and then that tag can get damp and pen can leak. So maybe putting that in a plastic bag itself is helpful. And then we also keep the information recorded in a GPS system so we can refer back to it then. And there's many different types. You could get a GPS itself, or there's many different apps for phones. The other thing when you're recording an area is when we're collecting from trees, we're usually collecting from one tree at a time. We're making single collections. Sometimes that's difficult when it comes to shrubs and berries, especially with cranberry or blueberry. They are growing low to the ground and it's almost impossible to tell each plant from each other. So we'll take a general location of where that collection was made. For the post-harvesting, we want to try to keep the seeds as cool as possible. The seeds themselves and the fruit, the seeds will start to degrade if they heat up too much. So in the photo in the middle of the slide, it shows a collection of nanny berry that's in a bucket full of water. And that was because it was collected on a very hot day. And you don't just want to leave it in a plastic bag in the back of your truck or in the back of a car, because that can cause some severe damage to your seed. So soaking it in a bucket of water to cool it down a little bit until you can bring it into an ambient temperature environment is really helpful, as well as removing any twigs or leaves, any sort of organic material. Once it's picked, it starts to degrade and that produces heat, and we want to reduce that as much as possible. So when it comes to extracting the seeds from the fruit, there's many different methods to doing this. And it really depends on what is the composition of the seed. Is it hard? Is it soft? And how much you are working on. So the most common method that we use to extract seed at the Seed Center, because we're using smaller seed lots and seed collections, is we use a kitchen food processor. And we'll put it in the food processor for about 4 minutes before, again, removing twigs and leaves. And we generally start with a 1-to-1 ratio of water to berries. Now, you can change this depending on how much the berries float in the water, how thick of the slurry that's created, and how many berries you're putting in. So you might want to put more water, you might want to put less water. That's kind of a judgment call you have to make. I've made the mistake of not putting enough water, and I overheated the food processor, and then I had to wait for a couple hours for it to cool down before I could use it again. So that's something you have to keep in mind. Sometimes the berries are too hard to use in a food processor, so we actually mash, squish, or crush them. And you could use the back of a Nalgene bottle, you could use a potato masher, anything that would squish the berries down and basically get the seeds out of the skin. And afterwards, you would rinse it with water to remove any fleshy material. Now, if you're trying to work with larger seed lots, the food processor might not work. And this is where we would encourage upgrading to a DibVig, which is the machine with the green bucket pouring into it. And basically, the DibVig was created to extract seeds, and you can use it for larger seed lots. And you can go to their website and check out how to use that. There's detailed instructions on how to use it with different species. But this is what we have used in the past, and what a lot of nurseries that we work closely with also use to extract seeds. For the gold panning technique, it is one of the ways that we use to remove the fleshy materials. And we actually have a video that demonstrates this technique. It's a good way to remove the seed from the fleshy material by floating it in water and using flowing water and vortexes to extract. So we'll be providing the links to these videos that outline all these cleaning techniques and goes a little more in depth on how we do it, times, volumes, and techniques. For rinsing and drying, we want to remove as much debris as possible. We want to purify the seed as much as possible. So as Donnie was saying, sometimes the seed doesn't actually mature. Sometimes the seed doesn't get pollinated or fertilized. So you just have an empty seed coat. And a good way to separate those empty seeds from the full seed is just to float it in water. So you'd put your clean seed into a jar or into a bucket, and you would want to keep the seed that sinks, and you'd want to get rid of the seed that floats. But you want to make sure to do cut tests on anything that floats, just to ensure that you're actually getting rid of either parasitized seed or empty seed. So this is the drying room that we use at the Seed Center. We have these racks that have window screens on them. And basically it's a good way with an ambient temperature. We usually try to keep it at 25 to 30% humidity in here. And it's an important part for cleaning the seeds, because after you put in the macerator, or you crush it, or you put in a dip vig, there's often still a lot of fleshy material stuck to the seeds. And it's very difficult to remove when it's wet. So what you can do is you can put it into the drying room and you let it dry for 24 to 48 hours. And then you put it all into a bag and you actually just rub it. And then all that fleshy material will be dry and it'll flake off very easily. And after it's flaked off, you can use a sieve, you can use an aspirator, which is the picture of me on the right there, using one of the aspirators here at the Seed Center. And basically what that does is it blows air through the bottom and will separate the flake debris from the actual seeds itself. Sieves can help separate things of different sizes, as well as gravity separation, but that's a pretty advanced technique. Again, in our video, we have in our videos we have examples of how to do these cleaning techniques. So I highly encourage to go check those out. If there's any questions or so about seed cleaning or different species, feel free to reach out to us. We'd be happy to help. Again, this is a little description of what it would look like to clean and extract seeds in a more industrial scale. And I'll let Melissa Sparing take over from here and explain a little bit about this and continue.
Excellent. Thank you, Karen. Yeah, so hi everyone. I am a seed biologist and I help out with a lot of our outreach and training. And this example comes from the Ontario tree seed plant. They did document before they closed at least all of the equipment that they use. So the basic processes and steps that are here in the settings that they used. I mean, it's kind of like a recipe. If you were doing a large amount, they had a lot of large scale equipment and they had a custom made sort of dip vig machine, but same process. And it's just kind of interesting to me. If you get into having a fanning mill, it works the same as sieves. But it's a technique that's been around for a long time that combines both sieving and fanning. So when you're getting into large scale cleaning, you do need to, consider and think about scaling up your pieces of equipment, but they all work basically on the same principles. So that publication is available as a PDF. I'll see if it's posted somewhere. I don't believe it's posted on the Ontario government website, but it is available from the MNR or the MNRF. And yeah, we can help troubleshoot a few of those things, but it also emphasizes just documenting as you're building up your skills. If you're a nursery, really writing it down so that people really understand the steps is really important. Very few people write this part down. So, okay, next slide. And one of the things that we have in our arsenal of tools now, especially for problematic shrub species, but any problematic species, is If anyone's familiar with these diagrams from the drawings from the Woody Plant Seed Manual, they're extremely useful when you're doing cut testing to compare and see what a healthy baby should look like inside these seeds. Shrubs have very diverse morphology, and they have a lot of different functions, especially around the micropyle, or sometimes there's plugs where the root is supposed to emerge from that have to degrade. So it's neat to see them when you both cut them and then compare it to the diagram. But for us, our new X-ray has really been handy to judge seed fill at either various parts of our cleaning steps or at the very end. And even after we've done all the best that we can with water flotation, we can still get examples where we have sort of malformed embryos, like in the snowberry that's up in the top right there. The sort of slightly lighter seeds are probably not viable. So we could take a sample of 100 or multiple samples of 100 and get an average to correlate with our germination testing. So this service is available to anyone that needs some troubleshooting if it does not conflict with what might be required in your province for other folks that may be having this. But not too many seed centers have an X-ray at their disposal. So we're really happy to be able to offer it to folks that need to troubleshoot. Okay, next slide. And okay, so one of the things that is obviously the ultimate goal is to be able to guess in however many pounds of beautiful clean seed you have, how many seedlings you're going to get out of it. And it can take quite a lot of steps to go from your initial collection. So if you click again, Kieran. So your initial bucket to your cut testing, down to all the floating and filling to get down to filled seeds, and then figuring out how to break dormancy. And this part is very challenging for us. And as Donnie explained in our mandate, we didn't really start working with shrubs until, I think truly our first experiments in our annual reports started happening around 2009. We did some work with some of the oil sands group on chokecherry to improve our prunus stratification treatments in 2014 and 2015. And it can take a lot of years to sort of break the code. So we do a lot of experimentation, and then when we know that we can reliably get germination, we're able to sort of publish our germination guidelines. And that's another document we're going to provide everyone for at least the species that we have some assurances. And we do have a lot of dormancy breaking treatments that are different than, say, the Woody Plant Seed Manual. So that's interesting because there's not as many Canadian samples in that book that was used when they published it. Okay, next slide. And so, for shrubs, a lot of them are very deeply dormant because they have underdeveloped embryos. So they typically can take a year or two before they will wake up. So our biggest tricks really are mimicking the seasons. A lot of the early berry species, to put them immediately into cold storage, they wouldn't normally be used to that. So we tend to give them a warm stratification treatment first. And we have to be conscious sometimes too of certain shrub species that drying the seed down to our storage moisture contents to put them in the freezer may actually deepen the dormancy. So a lot of our treatments, we sort of keep an eye on the fact that they have been dried, and we may need to extend dried seed to more weeks instead of fresh seed that may germinate a little faster. We have rock tumblers. We use sulfuric acid sometimes, boiling water. There's a really great manual from the US. The US Tribal Nursery Manual has some great explanations of scarification and pictures in it for a lot of similar species, especially in the western provinces. And then, of course, our number one for almost all of our shrub species in Canada, they typically go through winter, so they need cold, moist stratification anywhere from a month to 4 months or longer. And then sometimes we do alternating cycles, so we will do 1 to 2 years of going back and forth between a summer temperature, a winter temperature, a summer temperature, and so on and so forth. So you can see through our annual reports, you can see how we've developed that. And then we always look at ungerminated seeds before we really give up. Doing cut tests at the end of your germination tests, if you have a percentage that are still firm and fresh, it means that perhaps the seed lot needed another cycle or treatment. And we have lots of species where some of the seed will start germinating at the 4 degrees Celsius step, and then we have to sort of continue to try and coax the rest of them along. So it's not always they don't always wake up in the same week. So there's one more bullet on that slide. And we don't use this as often, but we know that there's a lot more study that could be done on using gibberellic acid. Gibberellic acid can be used as a substitute for cold moist stratification for some species. And, I've seen references to 500 or 1,000 parts per million for shrubs, so that it can penetrate through those really hard seed coats. But it's not as well researched, I'd say. It's an area for research. Next slide, please. And yeah, we're, as Donnie said, and as our inventory shows, we've been branching into more shrub research or shrub samples in our seed bank available for researchers. But we still have a lot of species that we do not have many samples of, if at all. Sometimes we have 1 or 2 seed lots of one Amelanchier. So we need to stock up too. A lot of what we're hoping to do is, asking for samples or being able to buy seed that is excess so that we can put some away for the long term in all this activity that's around seed. So if you have some really good crops coming along your area, please let us know. And we will try to see if we can get some extra from you. Next slide. Oops. And I would just like to reiterate the fact that seed collecting is really fun. And I've had, I think when you can do it with people, we can do it with a crew, you can do it with your family. The thing about berry picking is that for a lot of groups, it's a cultural activity to go out and harvest wild blueberries or pick wild strawberries. And, if we treat it like food, then we often, if we save the seed from it, then we also are doing something that gives back to the land for what they gave us. So I would just like to reiterate that it's really fun, and thank the plants if you can. Next slide. And yep, we have a really great team at our seed center, and Lucy's our newest join. So Lucy did not have a headshot for me, but all of the folks that you see on the slide here, and Naren has actually just hung up his hat for retirement to move to BC. So those that may have spoken with Naren before, he has moved on, but will be a director at large for us. And this whole team helps run the lab, helps run our field seasons, helps run our training and seed orders. Okay. And I have the last poll here. So in lieu of all the things that you guys are working on this year, what are your biggest bottlenecks in terms of, achieving your goals, achieving your objectives, running your business, running your volunteer programs? What's kind of your biggest hurdles? And I'll leave it up for 20 to 30 seconds. I really encourage everyone, again, please ask some questions. Please speak up as to your problematic species in the chat or in the Q&A, and we will endeavor to address them. And there's a number of very good growers on the call and nursery folks. If there's anything you'd like to speak up to your experiences with this topic, we very much welcome into the Q&A if you can speak up. All right, I'll leave the poll up. There's still answers rolling in. 10 more seconds. Thank you. 65% participation rate. So this is good. All right, training is high, seed availability is high, profitability high. Yep. Okay. Thank you very much. All right. So yep, this is just all the citations, and a lot of this, we're going to write a Q&A document for this to include a number of the resources we shared. And if we skip to one in English, and then there's we found some additional resources in French. So if you have French speaking seed collection needs, a number of these may not be in print. You may have to get in touch with the Quebec Ministry of Forests, but they have reproduced a few seed collection guides, and some of them are really quite good on a number of other species. And the last slide that we did want to show, and I only wanted to emphasize this as Donnie mentioned it. So the next slide, Karen. Is just sort of the timing. And so this is an excerpt again from that Seeds of Ontario Trees and Shrubs. Full disclosure that I helped produce it, so I'm very familiar with it. But any guides that sort of emphasize when you should be looking, or if they emphasize which species are in demand for certain regions, I think one of the things a lot of folks are struggling with right now is how to line up supply and demand. If you're not working for your own nursery and setting your own targets, you know, it's often a case of finding out who's the local native growers in your or end users in your region, and then coordinating with them. It's always a tricky thing about working backwards from the time a grower needs the seed in their hand to make sure they can grow it on time. So I really, you know, a lot of what we're trying to do with the seminar series is connect people and have people emphasize sort of the dates that they need to know. So if someone's gonna, you know, say that they need to grow 100,000 cherries for a project or for a shelter belt program, you know, talking to the collectors with enough time so that you can line up all these things in a row before the seed disperses is really important. And there's a lot of really interesting shrub species that, you know, they might be minor in demand, but if you become an expert at collecting them or have some really good collection sites near you, you know, they could be a specialty that you work on. So I just want to emphasize that this manual is good at laying out sort of the start to finish things, including how to find, you know, buyers if you're an independent seed collector. But there's lots of different ways that seed supply is sort of happening in different channels that it goes throughout Canada. So there's no one perfect answer to any of this. And I just want to say this is, hope everyone will stick around for the Q&A, and please, you know, put your hand up if you'd like to share some things that are going on in your region and sort of make yourself known. So I think that's the end of the presentation, and we'll get to the questions if the rest of my panelists would like to unmute. Thank you very much. We did good on time.
Yeah, thank you, Melissa. We have a couple questions rolling in here, and please feel free to ask as many questions as you'd like. If we don't have enough time to get to it now, we will get you an answer at a later date. So let's start with the first question that I have here from Lindsay. She asked, what measures, if any, should seed collectors take, if any, to stay away from cultivars, hybrids, etc.?
Ooh, yes. So one of the slides that was a little hidden, I don't know if we can skip to it and show it, the one that had sort of the table with the number of introduced and native trees and shrubs in Canada. So shrubs are widely traded, and other than in Alberta, there aren't any rules that specify, there aren't any rules that necessarily specify using native shrubs specifically beyond that as a policy. But, you know, as you can see, there's a lot of different shrubs in Canada, 489 native species and 127 that are well introduced and naturalized. And even in the past, there's been a lot of species planted for, you know, so quote unquote wildlife enhancement or hunting. So there is definitely a lot of cultivars and hybrids out there. And the closer you are to settled landscapes, that's far more likely. In the prairies, there's been a lot of development of native cultivars, like say they might be a native service berry, but they've done specific selections for increasing the berry size, increasing fruit production. So I'd say if the goal is restoration, I think those are discussions that folks need to have with the nursery and the end user to say, you know, is there a tolerance for any particular hybrid? Some species and genre hybridize freely just in the wild on their own. So that's Crataegus and Melanchiers. There's a few other shrub genre that not coming to me right now that you will find natural hybrids, and someone may just label them as SPA in the catalog. So I think you have to ask every grower, and you have to talk to the project planners to know what they will tolerate. And in the 2 billion tree program, you know, the emphasis is on using native species, but there is lots of areas of Canada where there'll be introgression. And I think that's just where you need to be good at plant ID and try to give yourself maybe a kilometer from sort of cultivated garden areas if you're trying to avoid them at least. But there's not really a good answer as to isolation distances for shrubs because they're pollinated by insects. So it depends on how far the insects fly. Hopefully that answers it.
Perfect. Another question from Kristen. With many shrub species having a high tendency to hybridize, what process are you folks taking to ensure you're getting correct species? And do you have specific use or mandate to specifically target hybrids at all? So fairly in the same vein there.
I'll let Donnie take that one.
Yeah, so again, really good questions. And, you know, as Melissa said that, you know, we've been in the tree business for a long time and we have standards with our tree collections. You know, we don't collect within town or city limits. We make sure we're collecting from wild stands as far away from urban centers as possible because we understand that, you know, in our unregulated urban non-crown land sort of setting, there is potential for not only different species, but different genotypes from common species that have been brought in and planted. And, you know, historically, we're looking for our native genetic diversity. So it really is an issue. The National Tree Seed Center, we really haven't done historically a lot of work in the urban, I'm calling the urban center, you know, anything outside of crown and provincial and park lands. But it is something that we are considering. And so far, when we are making our collections or encouraging it, we do try to be as far away from potential contamination, genetic contamination, we'll call it, sites. Having said that, you know, in the urban setting, that, you know, we said that sort of last presentation, that if you have, you know, I think there's an understanding within the 2 Billion Tree program that in certain settings, you know, our non-native species might not do as well as some of the cultivars that are, you know, have been brought in and have been developed to grow in these you know, our urban settings are not natural settings. I mean, they're becoming natural over the course of history of man, but ecologically speaking, they're still not. So I don't think there's an easy answer to the question. I think it's almost species by species. But the fact that we're thinking about it and the fact that you're asking the question, it's good because it's one that we should be considering. What are we collecting, making sure, like, do we, and where is it going to end up? So if we're making collections and they're going into urban or more, you know, occupied settings, it might not be as important as opposed to if we're actually planting out, you know, riparian zones in the natural landscape. We wouldn't want to be planting cultivars in those particular areas. That's all I have. If anyone wants to pipe in on that.
I was just going to add one thing that I know from our history of our collection is the one thing that we have backed up where there was a specific mandate for breeding material was from the Agriculture Canada Shelter Belt program. We did back up that breeding program where they were specifically selecting certain species that are not native because they were trying them for hardiness on the prairies. And Bill Schroeder was very diligent about, you know, at least keeping track of where they all came from and all the crosses that they made for the prairie programs. So there, once in a while, there's a collaborator who will ask and say, can you back this up? And the Morden breeding station was working on trying to do some controlled crosses for, I believe it was, there's a blight that affects Pernis. And they were, you know, asking and inquiring with us last year, the Canadian Nursery Landscape Association, about research material that fit that. So it's kind of, I mean, sometimes people have even said to us, why don't we have a collection of seed specifically for invasive species so we can study the problem? So, you know, there's a lot of different needs that research programs have of our collection, and you know, we've had to adapt over time, or we make new we evolve from it. So our emphasis is on native species, but there may be needs sometimes for collecting non-natives or hybrids. And Kieran just reminded me as well, I'm going to share the links for the videos. I'm going to share the files for the collection or the equipment list. I would love if anyone on the call with the FAQ, with the question and answer period, if there's a nursery person or a grower that would like to speak up and talk about issues around shrubs or these kinds of species that things that are needing attention, that's always really good for us to hear. So we can unmute people if you'd like to speak live.
Yeah, we have another question. I want to remind people to put their questions in the Q&A tab, which would be located in the toolbar there beside the chat. Just helps us keep track of questions now and afterwards when we're doing the recordings. So Steve asks, would there be any value in pursuing genomic screening for individuals/populations that are located in more urbanized or agricultural areas, or available generally through suppliers?
Yeah, that's a question that needs, I think, it's a great question. It might be one that, Melissa and I might have to sit down and, if Melissa wants to take a crack, but I think it's one it's best that we sort of think about it and reply. And again, any answers to any of these questions that we don't feel comfortable answering at this time, or might just not know the answer, again, we will provide the answers to everyone participating as soon as we have the answers. Melissa, do you have anything on that?
I think there's always value if you ask the right questions and you're trying to solve a certain problem to use genomics. I can just say from dealing with Martin's situation, we do genomic screening for butternut and for a few other species in our lab as well. But you need dedicated funding, you need dedicated technicians, and you need to be answering I think a really important question in order to get the funding to stay there. So I know, and Steve, you do know about genomics, so if you'd like to unmute, you could share your thought as to what you think the importance would be. Is it about provenance? Is it about figuring out horticultural introgression into our native areas? So there's no funding program for genomics right now for shrubs specifically, not at the national level. And I'm just gonna go blank for a second while I upload files. So give me a second.
Okay, we have some more questions coming in. Awesome. This question's from Haley. GPS apps are mentioned earlier. Can you include the names of those in the email? Definitely. We'll do that. Another question. What are the best practices for Shepherdea canadensis, different than the Woody Plant Seed Manual or the NAIT? So that's a question about a specific species. I think that's another question that we can get you some more information on afterwards. Some of those are more complex answers than we can give right at this moment.
Yeah, and there's a couple of really good documents from the Prairie Shelter Belt program and University of Manitoba that I've discovered about that one. So I think it's just always good. I think this is a good point though with shrubs, is that they're very variable across the landscape in terms of breaking dormancy, or even some of the seed collection techniques might work well in certain parts of the country and not great in others. And there's certain examples I know of, like bearberry that grows in the fire-prone ecosystems in BC is much easier to germinate than bearberry from the eastern part of the country because of physiological adaptation in the plug that keeps the root dormant. So there's a lot of, it's worth grabbing, as you know, having good literature reviews of all of our species, so that you can sort of compare and then optimize it locally. So we'll get you, yeah, I'll definitely do some research, Natasha, for Shepherdea.
Yeah,
anyone else on the line wants to answer her, you guys can also type, participants can type answers to other participants' questions as well. So.
Looks like there's a question that was answered in the chat regarding changing identification if we find out there's a hybrid. And I think, Melissa, you had an answer for that one.
One of the things
the presentation will be available to watch after.
Sorry, Kieran. One of the things we addressed earlier in the presentation is seed source identification. And we mentioned how with forest trees, there's been a lot of genetic and genomic work done, and that same work hasn't been done with a lot of our shrubs. But even in the conversations that we're having and the questions we're seeing, people saying, well, they're seeing differences in how particular seed sources one region react compared to others, all which imply that seed source, although the genetic and the science hasn't been there, the anecdotal evidence certainly supports that seed source and adaptive capability of these plants is very important, and hence backing up the need for planting, the right seed sources back into the right locations down the road.
Couple more questions. Tanis asks, are there programs that include seed conservation efforts for native grasses or forbs in addition to trees and shrubs?
So not as part of this particular program. And that's one of the, one of the gray areas we're constantly working with Department of Agriculture. So when it comes to grasses and forbs, a lot of that falls under the Department of Agriculture and not under our current mandate. Having said that, there are a lot of grasses and shrubs and forbs that, Department of Agriculture, I'm not saying not interested in, but they don't have collections for. And we have worked with different species at risk with Parks Canada, with conservation groups. One that comes to mind is the, oh boy, help me out, the beach pinweed is one that we're working. It's an endangered species on the East Coast. We're working with Parks Canada on that one. But also Frobisher's lousewort is a parasitic plant that only exists in New Brunswick and in Maine, and we work on that. So again, it's not that it's not part of our current mandate, but what we find that if conservation groups or approaches and there's nowhere else they can turn to, and we can't find them a place to turn to help support them, then we can look at applying for funding for specific species and take them on as special projects.
Someone asks, with berries soaking in water, is it good to weaken skins before cleaning? Are there any species known to be damaged by an excessive soak time?
I would love to ask I'd love to crowdsource this answer. I've never had an issue with, other than water getting smelly and fermented, over soaking anything. Most berries are very tolerant to that because they're within fleshy fruits. So I don't know of any studies that I've seen where soaking was problematic. If anyone else has experience on the line, I'd love to hear that. Can potentially imbibe some species if left too long. Yeah, I guess the risk is either, yeah, if you're not intending to germinate things right away, that soaking too long can hasten early germination of a fraction of the lot, or it can it may, if your seeds are immature and you're soaking them for too long, you may soften the seed coat up enough that when you do any sort of dibvig, or even, you know, a plastic blade even in the food processors may still do a little bit of damage to seed that is not fully hardened. So that might be the only couple of cases where soaking would be perhaps slightly detrimental. And a couple of people had asked me even before the webinar started about different liquids that people might use for float testing. There's some literature with cones about seed maturity, about using ethanol or some kind of, you know, there's different we use ethanol cleaning for, say, our birches and alders to increase them, but with our berry species, we don't, other than the stratification steps, we don't normally use any other sort of chemical liquid to do anything. It's almost always water, and we really just want to flush, you know, even soap and water, just to flush all the chemical inhibitors off the seed coat, because there's a lot of, that's the other thing too with fleshy fruits, is that the flesh actually inhibits it from germinating, so you got to rinse it well. Was there anything else I needed to share in the chat? Yeah, Jess brings up a good point there, covering your blender blades with soft rubber tubing.
That was something I forgot to mention when talking about macerators, and it's probably the most important thing, so it's not good I forgot about it, is that using a plastic blade, or as I said in the chat, covering the blade. Because if you use a metal blade, you're going to make a smoothie. Using a plastic blade, you're going to extract seeds, and we're more in the seed business than the smoothie business. So
I did reach out to Melissa and ask her if we should mention it, or if you should mention it for the plastic blades. So it's in the chat.
Perfect. Plastic blades, very important. Well, I'm not seeing any other questions coming up right now.
There's one from Heather that's in the chat about discovering snowberry growing near your cemetery. And someone must have brought it over for one of the services. It was never there before. So Heather, VASCAN is a really great resource for people. You can go in and build a checklist. It can show you all the native species for your province, those that are introduced, those that have been, you know, there may be native to Canada species, but they've been moved around even within Canada in restoration projects. But there is a native snowberry in Ontario, and there is also a non-native one that is extremely similar lookalike and tends to produce more fruit. So it can be often confusing to decipher between the Western snowberry, and a lot of the ones that are planted out in horticultural production are selections of occidentalis versus albus. So that's definitely where you'd want a good dichotomous key to differentiate before you collect which species you are collecting.
Just want to thank Jessica for sharing a video in the chat on choke cherry processing.
That is also a great video. Yeah. Okay. And yeah, there's one of the other nurseries we've worked with. One of the other things they have is there's a bit of a liquid rubber dip that you can buy. I'll have to ask them where they got it from, but you can take old blenders, and you know, you can also reverse the blades. I've heard of people doing that. You know, buy it. One of the things I'll suggest too, that I've suggested in other webinars, is if you're going to get into doing a lot of this, don't use your food processor that you use for your own food for seed cleaning. Like, have a dedicated dirty one that you know, you won't possibly accidentally, you know, consume. There are some fruits that are not edible and may have some toxicity to them, so just make sure you are aware of that before you mix anything into your kitchen. Excellent. And on equipment, I'd love to ask the folks on the line, equipment that you're using. Is there certain pieces of equipment that you know, more people should think of having if they're doing bigger amounts, or some tips and tricks that you have in terms of handling, like from the field to getting it back to your your cleaning area. We've talked a bit before about you know, little digital data loggers that we use. And you know, I mean, cleaning berries really isn't until you're getting into that big amounts where that equipment was shown for like 11 kilos of cleaned Amelanchier seed. I mean, you don't really need to spend a ton of money to get into cleaning berries, but a dedicated Vitamix, yep. The one thing I love that was very quaint with Bernie, who some of you may recognize from past webinars, was the fact that, you know, when we started at our seed center, when we mean small lots, we mean pretty small lots. And when Karen says milkshakes and smoothies, we have these 1950s baby blue milkshake machines, and they work really, really well for really tiny seed and small batches, but they only do about a cup of berries at a time. So they're not efficient for high use, but they work really well, and they're just, I think they came from a yard sale. But the DibVig is worth, if people are thinking about it or trying to get some extra funding, the DibVig is worth, I think last time we talked to him, Donnie, it was it's over $3,000 US plus shipping. But I will say, from nurseries that have them, they're solid machines. Most of the ones that are around North America have been used for 20, 30 years. And the fact that they're still manufacturing them, they're a very simple build and they're pretty robust. So it seems like a lot of money to invest if you're going to be doing a lot of berries, but they really are an indispensable tool.
And they're also very multidimensional. I mean, we use them for berries, but they can be used for de-winging other seeds. Yeah, they're a good tool. And yeah. I think the manufacturer claims that there's I think his success rate is like 99%, as far as almost all the machines he's ever made are still out there, still working 20 years later. So
yeah, and as Kristen points out, you can use them for dry processing as well. So, you know, milkweed seed, for example, or I've used them before for smashing up tumbling. You know, you can use them for tumbling pods of certain things for redbud or yeah, any other dry fruits you kind of just need to mash up. So very simple. And I'm pretty sure Jessica also has a really great video she did showing start to finish low-tech extraction of Norway spruce seed using a DibVig to de-wing. So we de-wing in cloth bags, but you can use a DibVig as well without causing too much damage, which is great. And of course, if you're lucky enough to be, you know, have a provincial seed center or provincial nursery for you or elements, I mean, most of those folks know the bigger piece of equipment that they have and have them well tuned. So.
Good. Well, if there aren't any more questions, first of all, this is our first real tag team event. The first two seminars I went alone, but it was really nice to introduce some of our staff, including Mary, Kieran, Melissa, and you met Lucy given the introduction today. The next presentation date is
July 12th.
July 12th. And again, at that time, we're hoping to maybe introduce one or two more of the staff for you. And again, I think that's very important. Again, we really are available to be on call to answer your needs. If you're in the field and you're making collections, again, in our previous seminars, we talked about using iNaturalist or even, you know, texting us photos, and we can let you know if things, you know, if we think they're ready, we can walk you through cut tests and help you make the best collections possible. So again, thanks everyone, including to all my staff. Thanks for showing up.